Hey guys, in this video we are going to go over gene regulation at the epigenetic level as well as at the transcription level. Epigenetics, what are we talking about? With epigenetics, these are going to be that your genes are going to pass down the DNA that you have passes down to your offspring, but you can modify those genes. You can put tags on them and make inheritable changes to the gene expression. So you still have the same genes, but you can't turn it on, or it's keep turning on when it shouldn't turn on. So these can be inheritable changes on gene expression. They don't change the DNA themselves. They are not mutations like we talked about where A's and T's switch out or C's and G's switch out. These have to do with the histone proteins. The DNA is wrapped around, and we're going to tag those histones, and that's going to affect the ability for transcription to happen. So at this level, when we're messing around with the histones and not touching the DNA, this is epigenetics. And so we mess with it with chromatin remodeling or DNA methylation. So what are we talking about when we talk about chromatin? Remember we talked about how DNA was this big long strand. If we put all the chromosomes together, it was like 6 feet long. And so to fit 6 feet worth of information, we wrapped them around histone proteins. And those tightly coiled together. And those are nucleosomes, and all the nucleosomes come together and have chromatin. And chromatin is what a chromosome is. It's just really tightly packed DNA on top of histone proteins that's going to make the chromatin. So to get to that tightly packed DNA, when we undergo gene expression, we got to unwind it a bit. And that's what I was saying before, where if we unwound every gene that we ever had, it would never fit where it needs to fit. So it has to stay tightly packed, and we only unwind it when we want to express it. So we've got to unpack it, be able to get to the promoter region, the operator, and the genes that need to be expressed to do that. So here's our picture of our histone. And we have, there's 1, 2, 3, 4, 5, 6, 7, and the 8th one, you can't see it. But the histone proteins and then our DNA strand is wrapped around those. And I think I got a video embedded in here, and it's also in the textbook that tells you, how many nucleotides of DNA is wrapped around a histone, if you're into those kind of nitty-gritty details, but I don't need y'all to know that. What I do want you to know is we've already talked about DNA is going to be negatively charged. When we talk about it's got that phosphate tag off the end of it, phosphate, sugar, nitrogenous base. The phosphates are negatively charged, so DNA overall has a negative charge that we'll go through once again. We're going to biotech and gel electrophoresis one more time. The DNA has a negative charge, and that those histones are positively charged, so they stay together. If we modify those charges and tack on groups, we'll talk about methyl groups and acetyl groups, it's going to cause those, the binding to lock tighter together, or it can loosen apart, and then we can get to those genes and express them. And maybe we didn't want to get to those genes and express them. So we can lock them up together, or we can loosen them apart. Based on the charges on the histone and the DNA. So there's two things that you can do. You can methylate or you can acetyl. The video here it is, here on acetylation is talking about just that. So acetylation, you're messing with the all of the little histone proteins, and you tack on acetyl groups onto them, and that changes the charge of that protein. And with the acetyl groups, it's tacked on with, what was it, histone, acetylation, transfer, something, the HATs. They're going to put these acetyl groups and tack them onto the proteins. And then there was the tacks that pulled them off in the video. But when we tack on these acetyl groups, it changes the charge, and the DNA will unwind. And now we can get to those genes and we can express them. So here we are, DNA is now accessible. We can activate the genes because of these acetyl groups. And these are tacking onto the proteins, onto the histone proteins. That's going to open it up. Or we can tack on another type of functional group. We can tack on a methyl group onto the DNA itself, and that's going to make the DNA even tighter. And so if you wanted to get into that DNA and open it up and express it, you can't, because those methyl groups are keeping it locked up so tight. So these things are called epigenetics. When you are tacking on these things, they're not modifying, you're not mutating the DNA, you're not switching things out, but you're tacking on these extra groups, and they will either lock it up or open it up. And so these little tails, these tag-ons, these histones, the histone tail with the methyl group or the acetyl group, those can be hereditary. So here we can see acetyl group has opened it up, or the methyl group DNA is inaccessible. Those things are heritable, even though you haven't mutated the DNA that gets passed down. These tails, these tack-on groups, acetyl and methyl groups, can get passed down. And so how do you get these groups? Well, sometimes these things are in the environment, and these environmental changes cause your body to tack on these things. So maybe what you are exposed to and raised with, what you have witnessed and experienced in your life, changes your gene expression. Just like we mentioned before, the astronaut going into space, his gene expression is changed versus his identical twin brother who didn't go into space. So your life experiences can change your gene expression at the epigenetic level. And so this article here was kind of neat. It was talking about people that have experienced, not great environments versus people that have experienced loving homes. They may have these gene expression changes that are passed down. So even if, you think your kids are better off than you were from your horrible experiences, you may still have some changes that pass down to them anyway. So it's something to look into. It was a neat article talking about that, and also talking about how maybe we could be able to modify those changes and try and remove those tails if needed, if it's discovered that that's what's happening. So this is the epigenetic level of being able to mess with gene expression. Either you've locked up DNA and you can't get inside of it to unwind it, or you have purposely unwound it when it should have been locked up. So methyl and acetyl groups, epigenetic changes. Moving on to, actually, DNA is opened up and now we undergo transcription. So when we talked about gene expression before, we had the promoter, we've got the operator, right? And then we have all these genes, and all this together was called the operon. And we have the RNA pole is going to attach on to our promoter, and then it's going to go down the line. It's going to go downstream and express those genes. Before RNA pole can bind, we are going to have, for eukaryotes, we have transcription factors. So they are going to assist RNA pole in binding to the promoter area. So there's two types of transcription factors that eukaryotes need. There's general transcription factors, and then there's some extra ones. Sometimes we have some special ones, so special transcription factors. And they won't bind specifically at the promoter, they'll bind at other places. But the general ones are gonna bind all over this promoter. And once they are there, then RNA pole can bind, and RNA pole will bind and go through, and we will express those genes with general transcription factors. Eukaryotes need transcription factors. Prokaryotes do not. So here's a picture of some general transcription factors found on eukaryotic cells. And we said that the promoter region is going to have the ta-ta box here. And then here's our transcription factor. And then here's another, and another, and another. And we have a whole bunch. And then whatever this gene is that it's expressing has all of the transcription factors that it needs. RNA pole can bind. And now we can undergo gene expression. So there are general transcription factors that every eukaryote needs for whatever that gene is that is specifically trying to express. Then there are the specific ones. And so again, those aren't going to be necessarily at the promoter region. They may be in different places, locations outside of the promoter region. So here we have an example of some. There are some DNA coding areas called enhancers. And enhancers are going to be here, and here, and here. And they're going to have these control elements that bind to activators. And so these activators are going to be bound to the enhancers. They are a special type of transcription factor. And now those guys can meet up with all of the other normal transcription factors that were here at the promoter region. And together with the special transcription factors and the general transcription factors, we are able to express this gene. But we wouldn't have if we didn't have this enhancer region coded in the DNA earlier, further upstream than the normal promoter region for those genes. So the special ones are going to be located and attaching on someplace else, and they will help with transcription. Can you turn this off? Yes. Eukaryotes need transcription factors to turn on gene expression, and there can be transcriptional repressors that will bind to the promoter and act like a repressor that usually binds at the operator, and it will stop it, and none of the other guys can continue on. We won't have gene expression happen. So you can turn this off. So this is one more place where we can mess with gene expression. So we can mess with gene expression at the epigenetic level, trying to unwind the DNA or keep it closed. We can mess with gene expression at the transcription level, messing with the transcription factors. Do we have all the factors, or did we put a transcription repressor on there and now RNA pole is not going to bind? We're not going to go through gene expression. So this is one more way. In the next video, we'll talk about the last couple ways that we can mess with gene expression and regulation, and then we'll talk about cancer really fast.